Jeffrey, you've spent over 30 years working across mass spec and chromatography. How has your perspective on separation science evolved over time, especially as chromatography-free approaches have matured?
Yeah, so chromatography really, for mass spectrometry, was enabling in many applications, right? Mass spectrometry, being a universal detector, is a very powerful detection technology. And with the advent of things like electrospray and being able to flow an active liquid flow into a mass spectrometer, you really opened up the applications that mass spectrometry could do in the applications across the board: toxicology, clinical, food and beverage, environmental, and proteomics, metabolomics. That added separation technique really helped mass spectrometry out. And then in the implementation of that, chromatography has really played a big role in moving into high-throughput assays and stuff like that. It really did enable mass spectrometry, but what we're finding is chromatography is still complicated. It's still expensive. It tends to be in mass spectrometry routine assays, kind of the area where most of the service and expertise is required and much of the cost of operation is. And so people use chromatography. It's kind of almost like a necessary evil. But what we're starting to find with the advent of new technologies, it may not be needed for all applications. And that's what we're leveraging here at Bruker is we've introduced technologies like MALDI into routine assays and then acquired IonSense and DART to implement DART technology into workflows where the mass spec is very powerful. It has a lot of separation technique. Chromatography might be being used, but in actuality, maybe chromatography is not needed for that application, whether it be a screening or quantitative assay. So what I've seen is that chromatography's evolution really was enabling, but now there's challenges in the routine market where maybe chromatography for particular areas and particular assays and workflows will not be needed in the future. And that's really what DART is bringing to it. We're not going to take over every chromatography method. There's still reasons for separation, and chromatography is advancing as well. But there is in the routine market, we see a need, and this was proven by technologies like Biotyper, which is a MALDI-based mass spec detection, where we remove the chromatography and boom, it was adopted on a very large scale for a clinical assay. And we're trying to do that with DART. So I think that the evolution of chromatography is it's around to stay, but there are areas where really chromatography is not needed. There's other separation techniques and ionization techniques that enable the workflow to be much faster, more robust, less expensive, requiring less expertise and maintenance. And these chromatography-free workflows certainly apply that. And other technologies like ion mobility and alternative separation techniques are certainly exciting of what might be moving on to replace chromatography.
And you touched on IonSense and the commercialization of DART technology, and you just elaborated on some of the benefits from the chromatography-free approach. What problems were you trying to solve back then as CEO of IonSense, and how does that work still resonate today?
We were trying to solve the problem of, well, when you go to a general person that is running mass spectrometry, and especially, say, a CRO or in a routine analysis, which triple quads, for example, are the king at that, they generally talk about cost per sample, and they generally talk about speed of analysis, and they talk about the expertise and reliability required to run the instrument. And those were some of the things that we wanted to do. We wanted to make workflows faster, where we could cut a significant cost out of the workflow, everything from consumables to solvent waste removal cost to the expertise required to maintain and support a solution. So, I mean, it really always comes down to, we could say we have great data quality and all this stuff, and that's important. But really, at the end of the day, when it comes down to things like routine, triple quad analysis, it's really about identifying how can you help cut costs, how can you run it faster, and how can it run more robustly through the assays that they're running. So those were really the key pillars that we looked at, that we saw DART applying it to triple quad assays. We could really help in particular assay areas.
It definitely seems like an extensive list of benefits. I was curious to hear from you what excites you the most about the expanding universe of chromatography-free MS workflows.
Well, for me personally, it's changing things a little bit, right? I mean, chromatography is overused. It is not green. It uses a large amounts of solvent, especially in high throughput labs. So what excites me is we're moving to more sustainable workflows. That's key. We're in an area where environmental study and environmental concerns are becoming more and more important. My daughter is getting her PhD in that, actually. So, I mean, that's what excites me, is we're kind of changing things. We're bringing a technology in like DART to make a workflow more sustainable while we're cutting cost and making it more accessible to the masses. In other words, I also think by adding DART and bringing this, we're making mass spec, by removing chromatography and that complexity and cost and making it more sustainable, mass spec will become more accessible to more users and more applications as well, not just the ones we're focused on today. Mass spec is moving out in the field. You can't move a whole chromatography system into the field and have bottles of waste and chromatography columns, equilibration times. That doesn't work when you're screening materials out of soil or out of a river. And that's what excites me about it. And I think it's really about where can you put mass spectrometry, make it easier, simple, cheaper, more sustainable to run, but also put it in applications where LC and mass spec together really weren't the right choice for it. Even if it was better data, it really didn't fit the purpose and use, like a point of need application, just for example, or competing with other types of technologies today that may not have the data quality, but do have a superior cost of operation and ease of use. So we want to move mass spectrometry into that, and that's changing things. So I think that's what's exciting. I think that's really the exciting part of driving those workflows. And people are going to look at chromatography where it's required and improve chromatography as well. I think there's a lot of areas and opportunity to move away from chromatography where reasonable, but also advance chromatography to meet some of the needs in the future, because they'll have to. There's a lot of companies that depend on very large chromatography and consumable businesses. And we certainly see that DART can offer that for my business and what we're focused on here at Bruker. But there's also, chromatography is going to evolve as well. And I'm actually really excited about the advent of ion mobility and mass spectrometry coupled, because that's another area where it's a very fast, reproducible separation that could augment chromatography in the future as well, or really another orthogonal separation technique to complement chromatography-free or chromatography workflows. It's a really exciting area, actually. And we're starting to couple that with our timsTOF. We just launched the TIMS Metabo with ion mobility TOF, and we're coupling DART to get that increased resolution for more complicated requirements, and maybe in metabolomics and in other areas. So yeah, I think that's really what excites me, is that we're kind of forging and convincing people that, because when I talk to people, they go, yeah, chromatography free, that'd be great, but I don't know. And we're proving it day by day. We're understanding the issues we want to solve, we're fixing it, and we're showing people that this is actually a possibility and this will move forward. And we're very close to really getting that spark of oh yeah, this really does work, much like what we did with mold in the Bruker group years ago.
Yeah, and as you may have noted, I've named this podcast concentrating on chromatography, but that's mostly for alliteration purposes, and that my company does the sample concentration after extraction primarily for LCMS and GCMS workflows. So I do really focus on separation science in general on this podcast, and that's why this is such a great topic for all of the listeners. But let's start there with chromatography in terms of where is it still essential, and at what point do you see it becoming more of that bottleneck rather than the benefit?
I mean, chromatography is still essential. If you're running a 900 analyte, pesticide analysis out of food or drinking water, you need chromatography. So very large, very complicated separation challenges. Chromatography is king and will continue to be required for that. But if you're looking at things that are a panel of analytes for screening, say you have a panel of drugs and you're screening those panel of drugs. Today, chromatography is long, expensive, and really doesn't compete with, say, like an immunoassay. If you can remove chromatography and still look at that panel, you don't need that chromatography. So that's where chromatography kind of falls off. And then if you add potentially ion mobility, you open up even more separation capability. But I still think with those things where you need to look at complex matrices, complex mixtures of compounds, you're going to want to use some combination of mass spectrometry, chromatography, maybe even ion mobility. Again, metabolomics is a really good example. You're looking at very complex things. We have a partner that we acquired, Biocrates, that makes a kit with 1,000 compounds in it. Well, that's not appropriate for that. That is going to need a very extensive, long chromatography to separate those out. And they do that really well. But what we could do is once you run that and look at it, use that in your discovery, and you find a panel of molecules that are appropriate for routine high-throughput assay, then you can move those to DART. You may not need chromatography to do that. You might need sample prep, which is a form of chromatography, right? So, but we really get rid of that middle part of having an HPLC column in a solvent workflow. So yeah, I think that's really where chromatography is king. It's not going to change. There's going to be more advances to make that better throughput and everything in the future. DART really fits into once you've kind of looked at that complexity and you've found your couple of, in a small molecule biometabolomics experiment, you found those or cluster of biomarkers that a small molecule, DART might be a really good high throughput analysis strategy.
So I know that instinctively labs might always feel like separation is necessary. What misconceptions do you see around the chromatography-free approach to mass spec?
Oh, that's a good question. One of the things is that you need chromatography to get rid of matrix. We used to advertise DART when we used to analyze materials and bulk substances and things like that. We could advertise we did no sample prep. And if you were doing an LCMS, you'd have to extract and put that in dissolve in some kind of liquid so you could analyze it. And DART didn't need that. You could just put solid compound into the DART and boom, you'd have really nice analysis. You could put everything from a leaf to a piece of paper. A dollar bill used to be the old demo, where with LC you'd have to do extraction. One of the misconceptions is that now that we're moving into biologics and very complicated matrices that these analytes are sitting in, depending on the application, we want to make sure that people understand we do still need sample prep and we depend on sample prep, right? In many ways, chromatography is a sample prep, is being used in high throughput triple quads. It's just another dimension of sample prep. And I think what we find ourselves proving is there's many applications where we can still minimize sample prep but use DART without chromatography, right? And sample prep with DART and the separation power of the mass spectrometers is enough to remove matrix and get an assay that's robust, runs sample after sample after sample. And that's what people think about is, well, I need chromatography because I need a peak, I need to separate out the matrix. But you can do that other ways. You can do that through easy, simple sample prep techniques that we couple with DART. And where that's needed, or you can do other things with DART that people don't really think about. Like DART actually is a separation technique. We use volatility. So we put a sample down on our mesh, we call it, and then we hit it with a gas, usually heated. And what we can do is we can do some separation by just changing the temperature of the gas while we're analyzing the sample and do a thermodesorption panel, which is in a way separation. So we can do things. I don't think people understand some of the levers you can pull with DART, coupled with a good upfront lab practice to do a simple prep, much like you do with LC or GC, same sample prep techniques. There's no invention there yet. But yeah, so all we're doing is removing. And I think the misconception is that the old conception is I don't believe you because you always said for years you didn't need sample prep for DART. And no way in biological fluids, I'm going to have to do something, whether it be simply a dilution or liquid-liquid, SLE, SPE, some immunoaffinity, something. And they're right. If we're coming on a biological matrix, we are looking at dilute and shoot type methods, just like electrospray. But we are also looking at minimizing and having very simple, fast, paralleled sample prep to be able to get what people are concerned about is how clean is that sample so things are mass specable and we're clear, we're differentiating molecules. And what we're doing is a lot of the applications that we're proving to people that actually it works is we're working on things like clinical research applications, I'll call it, because we don't have a clinical diagnostic yet, but we work with a clinical diagnostic mass spec company that we acquired, Recipe, where we're taking historically chromatography LCMS kits and we're moving them over to DART kits. And we're doing full validations with patient samples to show that the data is equivalent. And that's really how we're proving it. So people kind of got to sit back and say, oh, you're removing what we consider a very important part, and it is. But for many assays, once you move to high throughput and you have the power of MRM, you have the power of sample extraction when needed, you can bring those together and get enough separation and isolation of your needed molecules to be able to do a high throughput quantitative assay or screen. And we just have to prove it. They just have to let us in and give us a chance to prove it. We are submitting kits for regulatory approval in Europe, and we are moving forward on that with DART and no chromatography, and it looks brilliant. We're working with a lot of KOLs. That's the thing, is it is new. We're removing kind of something that people feel is almost that is required, but really in many assays it is, but in a big enough market to make it worthwhile, it's really not. And small molecule drug analysis, TDMs, there's a lot of capabilities there to do it. Screening of contaminants out of drinking water. There's all these things where really chromatography bogs you down on the front end of those analysis, and you really may not need it. And I ask people, just give us a chance to prove it. We've got a lot of capabilities that we're building up and learning as we go forward with this. And it's on us as a company to make sure this technology is mature enough and we can demonstrate it. We're really at that point now where we can show that we're in the process of developing methods. We have collaborators that are showing it, and that's the biggest thing that comes is we really have to prove it.
I'm happy to hear about the progress, and I certainly hope that this podcast can help you spread the message to a wider audience. We're seeing rising case volumes and certainly new drugs, increasing levels of abuse, and a major backlog for forensic labs. What are labs struggling with most right now from an analytical workflow perspective?
Yeah, backlog. Oh my God. They are getting all these drugs in. They're also getting a lot of these what's called MPSs, new psychotic substances that aren't on the list. And the backlog. There was a really nice article in that was put out. I forget the name of the newspaper, but maybe it was the Baltimore Sun or something. And their crime labs there have implemented DART to keep up with that backlog too fast because GC and LC are just too slow, unfortunately, to keep up with the amount of samples that are coming through the door at these crime labs and these investigative forensics labs. So they are looking at technologies, and a number of labs have implemented DART for bulk and substance analysis. So when you pull someone over and they have 25 boxes of pills, and then each one has a bag, right? They have to analyze every single one of those because every single one of those is a charge, right? And then they have to defend it in court and all that stuff. DART is being implemented to keep up with that throughput, but have the power of mass spectrometry to be true that is what you're analyzing. So really, yeah, it's unfortunate, but it is the amount of samples that are coming through the door and the amount of new substances that are coming through the door that are really challenging. And we work with NIST, for example. They develop methods on DART. They're very well published on it. They really are the leaders in DART for drug seizure applications. And they use our technologies for that and have been a wonderful collaborator with us as well over the years, from IonSense now into Bruker. and they do a lot of publications and presentations on using DART for increasing the capabilities of getting through this massive amount of samples for drug seizures. And states have picked this workflow up as what other federal government agencies. So really, yeah, it's just this massive amount of classic drugs, non-prescriptive illicit drugs, and new classes of compounds that are coming through the door that you can't develop a new chromatography or immunoassay for every single day that they come in, because it's swimming. So DART's nice because you just put it on there and run the bulk drug, and very nice drugs light up at DART. DART loves drugs. I don't know if I should say it that way, but anyways. But yeah, so I think that's the thing. It's just the keeping up in the backlog of samples is a challenge for these labs, and DART certainly helps that. And NIST has been a great supporter for drug seizure analysis, and we have collaborations with other large forensics groups that are looking to start incorporating chromatography-free now as well, to just deal with this backlog of both screening and quantitative analysis of drugs. And I think DART is certainly being very seriously looked at for just this large mass of increase in drugs, unfortunately.
Based on your description, obviously speed helps knock through the backlog at a faster rate. That's pretty obvious. But what you said also makes it sound like the second largest benefit might be the flexibility that those labs receive in terms of being able to use DART as a very flexible approach. Did I understand you correctly?
Yeah, absolutely. Again, when MPS comes in that doesn't have a chromatography method yet and doesn't have an immunoassay to screen, you can just put it on a DART and analyze it. And that's how NIST and other forensics toxicology labs use it for MPSs. Yeah, I think it's also the ease in enabling DART or mass spec to go other places. One area that we're collaborating with NIST on, and they've recently published and looking to expand a program. They have a program called RADAR, which is a DART program, where they have methods and capabilities to run drugs on DART in different locations. One of the areas that's really exciting is expanding where mass spec can go, is using DART in the field, in mobile labs, vans. Very cool stuff. And so we're collaborating with DART also now, not just to have these fast things at each of the crime labs, but able to move mass spectrometry in the field and bring basically lab quality testing to the field, which is really the next revolution, or one of the next revolutions of mass spectrometry, to move into applications that hasn't really been successful yet, which is moving into the field. There's been a couple good first steps in that thing. In moving certain types of mass specs, mainly low-performing mass specs into the field, because they want to be small and stuff like that. But moving lab-based performance into the field is really one thing that we're collaborating with NIST on and others for drug seizure, food testing, environmental testing, and we have a number of groups working with us on that now. And it really is enabled by DART, because they couldn't put an LC in the back of a van and run it around with solvents and all that stuff. So it really is kind of cool of where we're starting to put DART systems. So I think it is the versatility now that expands and opens where mass spec can be used and who it can be used by. It can be used by someone on a construction site. It can be used by someone in the van ultimately with someone that isn't necessarily a PhD mass spectrometrist or a chromatographer.
And there's certainly a lot of value in that. I'm sure people who are introduced to DART MS can understand it as chromatography-free screening. It's like the largest challenge you have in introducing this approach to forensic labs is really just establishing that confidence of a new approach.
Yep, absolutely. And it always is that way. My whole career, I've launched products that are new technology. I've had a lot of fun doing it. It's not as easy as taking on a technology that's kind of adopted and you kind of build on that. I've really been lucky enough to say, this is cool, let's try this and throw it against the wall. And yeah, you really have to work with the people that have the time and the mission in their facility to look at new technologies. And we get a lot of quick feedback an uptake in those labs as we start to mature. But now with those work that's being done, and then it's been really nice, some nice publications coming over out of the last few years, is Bruker now for quantitative DART, right? Because DART was always used as kind of a niche for high resolution mass spectrometry. That was 90% of the applications, qualitative identification and characterization. But when we joined Bruker, we really moved it into quantitative triple quad analysis. And that's really was the new thing. People knew DART worked. They saw it. Everyone had one in the corner on their old or new QTOF or high-res instrument. But people didn't have it on a triple quad. People weren't running biological fluids on it. And that's really over the last few years what we've done at Bruker. We've integrated into a triple quad. We've made a full triple quad workflow. We're automating that and buying things like kit companies to really augment and really prove that this is the future.
I'll tell you, as far as from the approach of someone who was totally new to DART before reading about this, I was totally blown away by the numbers. With LCMS, maybe you're running roughly 100 samples a day versus the 1,500 you might be able to push through with DART MS.
Yep.
Could you elaborate a little bit on what you've seen, how you've seen this improvement play out when it comes to, we touched on backlogs, but obviously lab staffing and just case throughput in general?
Yeah, obviously where DART has been adopted, your staffing as far as the operation of the instrument is a bit easier to get at. You don't need to have a person that is fully versed in troubleshooting chromatography. I mean, that's really the challenge of chromatography is when it's not working, you need someone that knows how to troubleshoot chromatography. There's a lot of moving parts, and there's a lot going on with the ion source, and there's a lot. I mean, it's a liquid flow into a vacuum with a lot of salts and dirt and stuff like that. A lot of people don't know this about DART, but DART has the advantages. We don't ionize salts. We don't ionize proteins. So when we do, and it is truly a small molecule analysis tool, we don't get a lot of the suppression effects that electrospray does. That helps us with not needing chromatography, because a lot of chromatography, the initial steps are removing macro. If you're looking at blood or serum, you're removing macro molecules, right? Larger molecules that love to ionize versus your small molecule. We don't deal with that because we don't ionize. Proteins and peptides aren't volatile, really. And so we don't volatilize them, we don't ionize them. So there's some things about DART, nuances about DART that you need to play around with or understand why we're able to do the things we're able to do in quantitative analysis versus doing shooting no chromatography with electrospray. That is, it has a lot of ionization competition issues where DART may not have that in many applications, which really gives it an advantage as well. So yeah, I think you're going to see your service rep less with a DART assay because there's no moving parts of the mass spectrometer. It's always the chromatography system. But anyways, I do think that you'll see in your operations lab requirement of less expertise on the front end, the ability to run things faster, troubleshoot things easier, and just get things through the lab. I mean, there is a bit more focus on, now that we're doing this into getting into high throughput automation and how does that automation work? And that's actually, we're doing a ton of work on automation and things like that. Not too different from high throughput MALDI type of automation. Very, very similar. I kind of look at MALDI as the chromatography-free protein analysis and DART is the small molecule chromatography-free analysis. So Bruker really does lead this market in designing workflows where they're adoptable because they don't use chromatography.
No, I think that's a great way to think about it. And I think you've done a really great job emphasizing how DART can simplify workflows in general. In terms of how certain steps may be lessened or disappear compared to LCMS, was there anything there you wanted to elaborate on in terms of what steps might be reduced in terms of how you actually simplify that workflow? Obviously, you touched on how sample prep is still part of the process, but it might be simpler.
Well, I mean, what's nice in labs is things go in plates. You can parallelize sample prep. So I don't see sample prep as a big bottleneck in the assays we're developing. It's another big thing. It's actually not related to DART so much, but it's our thinking and what we're trying to do to own the whole solution. Because the other thing that's changing is mass spec is moving away from a component sale. It used to be that it was okay to buy your mass spec from one vendor and your LC from another vendor and your reagents from another vendor and your software from another vendor. And that's starting to go away. In the routine analysis markets, you really want single support and make things more efficient that way. So one of the things we did is we acquired a couple companies that do kits. And I think that's one thing that also in the routine will help is providing more validated robust methods, which is the work on our side with DART. We really have to get those done. And we validate it around minimizing those things. But what we also do through a kit company is we supply the materials that we also guarantee and validate it. A really good example is we acquired a small company called Pinpoint, which is a forensics toxicology, clinical research toxicology group that basically supplies custom kits for people's drug analysis out of biological fluids for LC. And what they do is they remove all the inefficiencies of having to source 120 drugs for an analysis, right, that you run for drug confirmation. And they just make a tox box where everything's in that box that you need. You just add the sample and you can run it. So those are the conveniences also that are really important. Besides chromatography-free, we're applying that concept now to DART as well. And we're starting to develop ToxBox. And I really think it's something people should check out because people open their eyes. It's like, oh, you mean I don't have to go to, I don't know, this company and source all my drugs and then have technicians that put all this together? And no, you just get ToxBox and that's everything you need. And you add your sample, put it in the auto sampler and run. It's really cool. So those are the things we're also building in, because I think technology on its own is not enough nowadays. It really isn't, especially in kind of in the routine markets. They want something to come in, robust, get running it. And so we really, I think, have made over the last 2 years a couple very strategic investments in not only technology like DART and ion mobility and all these cool things that Bruker brings to the table that we're moving into the routine, but we've also brought in the expertise to make that workflow routine. And I think that's one thing. We talk about removing things for DART, but really, at the end of the day, the value comes when we support it and supply what customer needs to make the operations simpler as well. And I think the philosophy of DART and philosophy of what we're building here at Bruker with, say, drugs of abuse testing with DART on a triple quad, with the tox box and recipe solutions for drugs of abuse and therapeutic drugs that we might analyze in a forensics or clinical research setting. Those are really the things that add value as well. DART on its own is nice. It's a technology, but we're starting to do demos down at Pinpoint for drugs of abuse testing. We're doing demos in LCMS clinical assays that they develop the kits for. We're doing demos and showing people that DART works on these assays, validations on these assays. So I think I kind of expanded it on it, but it really is important to say it's not just DART, really. It really is our approach of bringing everything together and owning the solution. And removing chromatography just makes it easier for us to own the whole solution because it's easier and it's one less thing to worry about for customers.
I think you've sold me on doing one or more episodes on kits themselves and how that's really a game changer. Like I referenced earlier, I believe it was at ASMS last year where I went to a breakfast and learn, let's say, by Biocrates, and I was blown away by the work that they were doing.
Yeah, no, they're another group. Our life science group acquired them, but a lot of their work's on triples. And so we're collaborating quite a bit with them now. Yeah, no, they really do. And I see kind of the string of where they can help with researchers and metabolomics and lipidomics, but then we can pick up the kitting of the actual assay that's developed out of that panel, the subsegment of that assay for DART out of that panel. Yeah.
Yeah, no, I'm excited to learn a a lot more about kits in general because it could really revolutionize a lot of workflows. And one thing you touched on, whether it's consumables, solvent use, waste disposal, there's a lot of hidden costs, let's say, involved with traditional chromatography workflows. What do you see changing in terms of overall cost of ownership for labs in general?
One of the things is once you move to kits, the opportunity in some of the markets is move to a cost per sample model or reagent rental models. That was tough for mass specs in the past. And you see, actually, this is the way some of the big guys, there's a lot of excitement going on with mass spectrometry in, say, clinical diagnostics. I think that's the way mass spec's going to move. I happen to think the way we're doing it is better, but we'll see. We don't really compete with that because we're still living in more of the specialty labs. But yeah, no, I think it's exciting that the business model for mass spectrometry will change more substantially. And adding kits, for example, makes that reproducible, measurable. So you can open up a whole bunch of things that in a more research mode would be tough to do on that. And I think certainly driving down and making mass spec maybe not even a list price change. That doesn't matter. Really, the cost of operation and focusing on that as well. Bruker is never going to have the cheapest mass spectrometer. I shouldn't say never, but right now today, we don't have the cheapest. But I think with the way we're approaching things, we can demonstrate with DART a very significant cost of ownership benefit of the instrumentation. I think that's very important in the routine markets and really one of the key competition things that people look at. It's moving less and less from brand, where where it's getting more into what is my cost of ownership, how am I going to make money on this instrument, and how am I going to satisfy my customers, not my customers, but my customers' customers. How am I going to? This is what they're asking themselves. It's how am I going to satisfy my customers, and how am I going to do this robustly, and how am I going to do this where I actually can do it in a profitable manner. And certainly strategies of reagent rental and all these things we can put together with technologies like that to just cut the cost are things that people are going to look at, and they're starting to look at, and they have been looking at. But I think that could become more the norm than the thing. That's one thing. The research mass spec market has been challenged a little bit by the slowing down of grants and all this stuff. So the good news is in the routine market, they're still going to run samples, they're still going to run drugs, they're still going to run environmental samples. But how can we make for industry and academic and government labs a good, affordable, high reliable instrumentation and workflow for them to get done what they need to do in the most economical way? And they don't have to buy the cheapest instrument to do that, but they have to buy a workflow that meets that need. And that's what I mean with DART.
I had a great conversation on a past episode with My Green Lab, focused on how sustainability is becoming much more of an actual driver during the purchasing process. How do you see DART and chromatography-free workflows supporting those Green Lab initiatives?
Yeah, just think about it. So I walk into labs that have 50 triple quads with 50 LCs running, say, let's just say 400 samples a day on a high throughput instrument, right? Running 6-minute gradients, running a mill a minute of acetonitrile hazardous waste through their systems and generating liters and buckets of hazardous waste. That's gone with DART. It's gone. We use solvent, obviously, methanol acetonitrile with extraction. That is a infinitesimal fraction of when you're running 800 to 1.2 milliminute gradients for high throughput chromatography on your bank of triple quads. It's immediate. So you walk into a pharmaceutical company that's doing drug discovery, again, a very simple assay that you could develop on DART with no acetonitrile gradients at all. So that cost and the sustainability goals of that lab have just been met just by implementing a new technology. Now, I know it's not that easy to implement a new technology, but that's really the payoff of DART for these high volume labs is you walk in, you say, minimize the organic solvent waste. And let's remember, availability of acetonitrile sometimes has been a challenge in the past, depending on the industry. Cost goes up, cost goes down. But at the end of the day, you're using, 5 mils an assay, say, or 4 mils an assay for 400 samples for 50 mass spectrometers. I mean, that's just generating massive amounts of waste. So, and that's a cost for pharmaceutical and clinical labs. Clinical labs aren't used to that because they generally don't use acetonitrile at that level, chromatography at that level. It's just, and that's one of the challenges with mass spectrometry, LCMS, is putting these consumers of waste into a lab that generally didn't do that. It may not have the facilities to deal with that level of hazardous waste. That investment, when you bring mass spectrometry in, it's an investment just to deal with what those things produce, both facility-wise and disposal-wise. We don't have to deal with DART. It's a huge cost advantage of DART that we don't even sometimes talk about. We should talk about more. I got to put that into my sales pitch now. But yeah, so I think that's really, think about it. Think about how many mass spectrometers Joe Blow Clinical Reference Lab has, or how many mass spectrometers Joe Blow. I've walked into these labs and they have, a facility might have 100 or more. So yeah, it's very exciting. We have to get confidence though. It has to work and all that. But to me, once we prove to them this works, the investment should be there to drive this technology to a better place of sustainability and cost savings. By the way, it's also a much easier system to operate where it works. Maybe you cut down your chromatography from 100% to 10% because there's still some assays that you might want to use chromatography for, something like that. Again, I don't want to promise it for everything, but it's a high percentage and well worth it. If you're running your Biocrates thing or something, to panel of 1,000 compounds, that's not DART yet. You still will need a couple chromatography systems, but you don't need 100.
Yeah, no, absolutely. And I wanted to touch on immunoassay method, which traditionally those established methods work well, but they do produce high rates of false positives.
Yeah.
Where does the chromatography-free method come in, and for that specific issue, do you see an improvement?
Yeah, so mass spec has been trying to go after screening for a very long time and fails. And us mass spec people scratch our heads because our data and capabilities are so much better. So we've come up with some really good publications with our collaborators. Joel Kari at Yale had a really nice publication on opiates and versus ELISA-based assays and things like that. And a really great feasibility study for DART. Did it 4 years ago. He did a great job on an old instrument. We've come a long way since then with Joe. And what he showed was it's this thing good enough that us mass spectrometry people have to learn is with screening, we haven't beat immunoassay because immunoassay is good enough. But they don't like it because it does have a lot of cross-reactivity. You can't react to the drug world, new analytes coming onto the scene on the street. You have to put it on a mass spectrometer today, but it's LCMS, so it's slow and screening, a lot of false positives, right? So I see us coming in with mass spec, which people want to do. We just have to meet the criteria that immunoassay has established, which is simple, fast, cost-effective. Right? They've established that. And mass spec has not really been able to beat that yet because of chromatography, in my belief. And if we can remove the chromatography, bring that cost down, bring the ease of use down, and make it better, or ease of use up, and have a decent cost of ownership against an ELISA-based assay, we have something, for sure. And we can, there's really a couple really good examples where I see DART really playing a role is like MPSs. There is no ELISA. There is no affinity for. And the minute you develop an ELISA for it, it's not on the street anymore. There's 10 more, 20 more, 50 more new analytes, MPSs that have come onto the street in that area. So how do you deal with that? And mass spec is the way to deal with it. DART mass spectrometry is the way to deal with that. There's been some very good talks we've given with CFSRE, who focuses on this problem. They're a really important institute in looking at how we can meet these needs that are coming so quickly at us. And yeah, I think that's really an important fact of the technique and where we're going.
I always like to ask at least one question about the future. And I suppose the easy answer here will be that you'd like to see increased adoption. But Jeff, 5 years from now, where do you see this technology having the largest impact? Is it forensics or clinical research, or what are you most excited about?
Well, it's the areas my group is focused on today. And I'm going to lump a couple things. There's two key areas that we are looking at. Drugs, so drugs of abuse and therapeutic drug monitoring, as well as a cluster of vitamins and other things like that. But basically, moving DART into clinical applications, it's why we acquired Recipe. It's why we are taking LC-MS clinical kits that are IVDR and moving them to DART, right? And focusing on that. So I think we will have an established our value in that market within the next couple of years. And we'll see the adoption ramping up for both quantitative assays, but I really feel that if people are doing drug screening and challenged with these new entities that are coming into the market, DART is a good place to start to be able to deal with these things coming in and look at them quickly. I also think the others, and that's really a challenge for society. Drug testing, diagnostics testing is increasing, right? More and more diagnostics. Because ultimately, if you do the right diagnostics, you will help healthcare and cost of care. That's the theory. That's my theory. So I think testing will always be there. And then the other thing that we're dealing with as society, I mentioned my daughter's involved, is contaminant analysis. And I think the fact that you and I are polluted with microplastics and PFAS and heavy metals is an area to focus on and help humanity as well as find a business opportunity. And I think I want to see that Bruker, with these, not just DART, but ion mobility technology, the acquisitions that we've made in companies like Pinpoint and Recipe and Biocrates to really drive and support this growing number of samples and be able to monitor health and influence what happens based on the technologies we're providing researchers and people running mass spectrometries in routine analysis to solve problems. And we can do that in an economic way so it can get adopted and stuff like that. So that's kind of what I see over the next few years is these core areas of drug testing, both in the clinical and forensics world, as well as contaminant analysis, which leans over into health as well, right? People are looking at these materials in humans now to see how they're affecting us. But they're kind of almost interrelated of this. Basically, you could look at drugs as contaminants in our body as well. I believe that DART will have its place in those assays over the next coming few years. That's what I'd like to see. I think we have the right people working on it. We have the right collaborators giving us feedback and telling us what's working and what's not working. I think we have all that means we have the right focus, and that's what we're focused on.
Whenever I'm talking to someone new and discussing my work in the commercial side of science,
yeah,
I always focus on the value that comes from working on real world problems and being able to add that true benefit, which, not that I had a lot of experience, but as someone who worked in different industries, I see the real world problems we're solving in science as the most rewarding.
Yeah, absolutely. Like I said, I've been very lucky to ride a few technology waves and introduce new technologies. It's always been in the life science world or in the health world with mass spectrometry and chromatography.
We've covered a whole lot of ground on DART. I'd love to hear at this point, is there anything we've missed that you're just dying to tell people about who might be hearing about this for the first time?
I would love people to investigate more. Always love to interact with people and have people poke holes at it, but also, see what we're doing and see if it actually does fit within their organizational needs, their focus of what they want to accomplish, right? And to bring new technologies in. And, that's really what I'm interested in doing has been the most fun is these evaluations and collaborations and working with people that are even closer to solving problems than I am. I'm of this really on the front lines, and giving them tools to fight the battle and see how they work is awesome, is always fun. And that is progressing greatly. So no, I just, if, people want to learn more or have an opinion of what they want to share of why they think what we might be missing or what they're excited about and what we could look at with them, I always love to having those conversations with people. That would be great. I don't think we've really missed anything. I think you could do a whole segment on ion mobility and where that's going in the world. I really do. And Bruker's leading that. I think that there's a lot going on in the mass spec world, a lot of capabilities and technologies coming onto the market, like DART, like TIMS, that are incredibly exciting and going to change the world. And that's really what it's all about. But I really think this is, besides the applications we're working on in these labs, being able to move something in that is solvent-free for the most part. You can't say 100% because we do use extraction still today that uses solvent, but we can use separation techniques, things like that don't use solvent. So I think that's really, a big driver as well. And I think once we establish that DOT will work for someone's application, that's the natural, and that's really where it will go.
Yeah, no, I'm excited to hear about that, and I certainly hope to have a follow-up discussion with you to find out how things have progressed. I'd love to follow up then, but Jeff, thanks so much for your time today. I really appreciate it, and I hope I get to run into you in person, whether it's in Massachusetts or at a conference down the line.
Yeah, absolutely. Let me know if you have any questions. I really appreciate the conversation. I love to talk about DART. I've dedicated a few years of my life to it now. So
thanks, Jeff.
Thanks for the time. Cheers.